Good afternoon and welcome to the E-School of the European Society of Gene and Cell Therapy. My name is Hildegard Büning. I'm professor of infection biology of the gene transfer at Hannover Medical School and the current president of the ESGCT. As you might already know, the ESGCT is a non-profit organization promoting fundamental and clinical research in gene and cell therapy, as well as in vaccine development. As part of our mission and because education is close to our heart, we launched the ESGCT E-School, an advanced lecture course. We thereby aim to provide a comprehensive insight on the various aspects and areas of cell and gene therapy. Following the introduction of vectors and cargos, which also included designer nucleases, we had an insight on gene and cell therapy of rare diseases. And now we are at the final part of our lecture series, which is dedicated to cancer gene therapy. It is a great pleasure and honor to welcome today Professor Stephen Russell from the Mayo Clinic in Rochester. He's the current president of the American Society of Gene and Cell Therapy. He is an MD and a PhD, and he dedicated his scientific life towards oncolytic virotherapy, the topic of today's lecture. More specifically, he aims to develop engineered oncolytic viruses that are selective destructive to cancers. Moreover, he is interested in elucidating the molecular targeting mechanism responsible for the specificity of these viruses, and to enhance advance the oncolytic viruses to clinical testing. With a great number of similar papers, he has advanced the field of oncolytic virotherapy, and we are very much looking forward to his lecture.
Okay, thank you very much, Hildegard, for the introduction. It's a great honor for me to be invited to give a lecture in this series, which I think is an outstanding series. Despite having lived in the USA for the past 21 years, I do still regard myself as a European. So this feels like coming home. So I don't have control of the screen, so I'll be asking Gail to advance slides for me. Gail, if you could, next slide, please. I do have a disclosure in that I'm involved in the company Virotherapy, which is developing oncolytic viruses. Next. So the goals today of the lecture that I'm going to give to you are different from many of the lectures I've given previously on oncolytic virotherapy. I want to review some mechanistic aspects. First, I'd like to, and I'll use VSV as the example oncolytic virus for this. But first, I want to review the two-step action of oncolytic viruses, which involves an oncolytic and an immune stage. I want you to gain an appreciation of the anatomical, geometric, and mathematical aspects of oncolytic virotherapy, which before you run away are actually pretty simple in my view and quite helpful when you think about oncolytics. I want to just briefly review the main barriers to systemic delivery and intratumoral spread of oncolytic viruses, and then briefly consider some approaches to virus engineering that may impact the boosting of anti-tumor immunity after oncolytic virotherapy. So we'll start with the mechanism of action. So this is a viral infection. Viruses, you all know, are obligate intracellular parasites. A single virus enters a cell, its genome is amplified, and its genes are expressed. The proteins required to make new virus particles are put together with the replicated genomes, and out come up to 10,000 new virus progeny. So it's that we're trying to exploit. Next slide. So we have a very large number of viruses out there to choose from. They come in all shapes and sizes. There are RNA and DNA genomes, and within both of those categories, there's a great deal of versatility depending on the symmetry of the capsid, which may be icosahedral or helical, and the presence or lack of a lipid bilayer membrane surrounding the viral core. And then there are positive sense, negative sense, single-stranded, double-stranded viral genomes. And, you know, this is really a beautiful array of nature's nanoparticles, and pretty much every class of virus in this slide here can potentially be used as an oncolytic. Next slide. So this is how oncolytic virotherapy works, and this is the two-stage action that I was talking to you about. So first off, the virus infects the tumor, and we have to get it there to do that. The infection then spreads in the tumor and results in the infection of multiple tumor cells, leading to their inflammatory killing and the release of tumor antigens. And in the context of this inflammatory reaction that's occurring within the tumor, there is an opportunity for the immune system to recognize tumor antigens, and anti-tumor T cells are thereby amplified. So you get a boosting of T cell immunity, which can then have an impact on distant tumors that have not been injected, or alternatively, if the virus is given systemically, can impact uninfected tumors within each tumor deposit. So you've got two steps here then, the oncolytic phase where the virus destroys tumor, and the immune phase, which is more prolonged, where the immune system mops up the remaining tumor cells. Next. So I'm going to use this virus as an example to sort of track through how oncolytic virotherapy works. This is vesicular stomatitis virus. It's a negative strand RNA virus. It's a rhabdovirus, so it's bullet-shaped. Very simple genome with just 5 genes, and we can insert additional genes into the genome at intergenic junctions. So this virus, VSV interferon beta Nis, has the interferon beta gene inserted to enhance the ability of the virus to inflame tumors, and the Nis transgene to allow us to track the propagation of the virus by non-invasive imaging. Nis is the sodium iodide symporter naturally expressed in the thyroid gland, which is the reason we can image the thyroid gland using radioiodine and other radio tracers. And one important point about VSV that I think needs to be recognized is that it's naturally a pathogen of hoofed animals, of cattle in Central America and the southern United States. It causes a self-limited illness, a blistering illness, and is non-fatal. Many farmers have been exposed to the virus and there's never been a fatal human infection. But because of its natural habitat, the seroprevalence amongst cancer patients is extremely low. And so we can look at this virus as a systemic agent for the treatment of cancer. Next slide. So here are some electron microscopy studies on this VSV interferon beta Nis virus. And really, I'm just trying to show you here that the bullet gets longer when you add new transgenes into the genome. So the ribonucleic capsid here wraps the genome to form this helical bullet-shaped structure. And the longer the genome, the longer the bullet. So in the middle here, we've got VSV with the interferon gene, and on the right-hand side is the virus with the human interferon beta and the Nis transgene. Next. So that virus, having been made, was then extensively studied by Shruti Naik. Shruti Naik, shown here, actually did an awful lot of the work that I'm going to be talking about today. She was a graduate student and is now a postdoc in my laboratory. But she tested this virus as a single intravenous injection in multiple tumor models. And I'm just going to show you some of the data from that. If you could advance the slide again, keep going. And again. And so here we've given a single intravenous dose of virus to animals with subcutaneous 5T-GM1 tumors. These are immunocompetent mice. And you can see that there's a very clear dose response. You might not expect that for an oncolytic virus, but it's real, and you can see it here, that we need to get up to quite a high dose before we start to see activity with systemic administration. And the higher we go, the better. And if we go to the next slide, you can see that if we take the dose even higher, we reach a point where tumors are completely eradicated and do not recur. And if you look on the left-hand side here of imaging data, this is in a single animal, imaged days 1, 2, 3, and 4 post-virus administration using pertechnetate, which is a NIS radiotracer. And what you can see is that the tumor is really strongly lighting up on day 2. This background signal in thyroid and stomach is just natural NIS expression. But the tumor strongly lights up and then fades as it regresses. And so we really are very interested to know then, for a mechanistic study of oncolytics, what is going on inside these tumors? I mean, what is the associated behavior of the virus within the tumor that results in this intense NIS signal, which then fades over time? So the next slide shows the first experiment we did, which was to give the radiotracer and then explant the tumors, slice them, and put them onto radiographic film so that we could see where the radioactivity was in the tumors. And you can see that on day 1, there are multiple small foci of uptake of the radioisotope. And then on day 2, you get the same pattern, but the foci are larger. So there's clearly an expansion of foci going on between days 1 and 2 in this study. Next slide. So we went on to look at that further, and what we did was to explant tumors that had been treated with a single intravenous dose of VSV interferon beta NIS at 6, 12, 18, and 24 hours post-virus administration. And we stained for the VSV antigens. And the green stain here is looking at VSV. And what you can see very clearly is that over this first 24 hours, there are foci forming and radially expanding as the virus spreads. And this was quite unexpected, that this was the mode of virus spread. You might think, looking at it, that it's obvious that the virus would spread in this way, but previous thinking had sort of brought in the concept of viremia and reinfection of the tumor. But in this case, it's just the initial infusion of virus infects multiple foci within the tumor, some of which expand to form these large infectious centers. And you can then start to think about, well, how fast is the virus moving here? And if you measure the diameter or the radius of each of these infectious centers in terms of the number of cells from the center to the edge, you can then, using the volumetric calculation of volume equals 4/3 pi r cubed, you can work out how many cells there are in each focus. And so if you look at this little graph at the bottom, you can see that moving from the 6 to the 24-hour time point, we're moving up to a diameter of something in the region of 30 cells per focus. And it hasn't stopped growing, by the way, at 24 hours, but that's how fast it's going at that point. And you can correlate that then on the right-hand side at the bottom to the number of cells per focus, which is increasing in this case to about 10,000, and which goes beyond that. So the next slide just drills a little deeper into an individual focus to ask, well, what is happening to the cells in that focus? Are they actually dying? And this is tunnel staining. And if you look at the red stain here is now for the virus, and the green stain shown in that blow up is the tunnel stain for apoptotic cells or dying cells within the focus. And you can see that there's a central area of apoptosis, and there's a peripheral area where the cells remain viable even though they're infected. So the infection is clearly spreading outwards. Next slide. And it's very interesting then, if you look at a slightly lower magnification, to see that this central area of cell death is then adjacent to a rim of viable but infected cells. And you can actually, by measuring the diameter of the rim, you can work out how fast the cells are dying after they've been infected. And the rim here, as you can see, is on average 10 cell diameters. And we could calculate from that previous slide that as the infectious center expands outward, each new layer of cells is infected every 2 hours. You can calculate that from the speed of expansion of the center. So if it takes 2 hours to cover that, if it takes 20 hours to cover that distance between the edge of the dead cells to the edge of the viable but infected rim, we can calculate that it's taking 20 hours for the cells to die after they've been infected. The other really interesting thing here is you would never expect that the virus would be traveling as fast as it is, getting to the next layer of cells every 2 hours. It's really moving much more rapidly than we see the release of progeny viruses from an infected cell in a tissue culture system. And you can see here that if cell death is lagging so far behind the leading edge of the infectious center, then clearly the virus is moving into the next layer of cells before the originally infected cells have died and released their progeny. So this isn't viral spread by burst, release, burst, release. This is viral spread by contiguous spread between adjacent infected cells, which I think is very interesting. So the next slide just takes a sort of higher magnification view of this. And again, we stained the virus infected cells red here. And we've taken tumor sections at 24, 48, and 72 hours. So we're moving out a little bit later. And what you can see in this left-hand panel is that after 24 hours, there's a lot of infectious centers. You can still discern them. There's some death of the cells in the center of these expanding foci. But by 48 hours, the foci are considerably larger. There's been a lot of them coalesce, and there's more dead cells in the center. And then by 72 hours, the whole tumor is infected, and there's extensive cell death. Next. So this is how we think it's working. So we give the virus intravenously, it extravasates from tumor blood vessels and establishes infectious centers within the tumor, which then expand radially with central necrosis occurring with a lag time of about 20 hours from initial infection. And then eventually these infectious centers coalesce and there's wholesale destruction of tumor cells. So next slide. So this is very informative to look at this model that I've just shown you. And obviously not all models are equally responsive to virotherapy, and we'll speak to that in a little while. But we took this model that we'd fairly extensively studied and tried to do a bit of mathematical modeling on it. Because all previous mathematical models were talking about the exponential growth of viruses, as determined in tissue culture systems, and weren't really looking at the anatomical, geographical constraints on the amplification of the virus and the spread of the infection. So if you think about what I've just shown you, there are really two critical things. There's the first, what is the initial density of foci within the tumor? And that's probably going to be a Poisson distribution if we assume it's completely random. And then what's the final size of each of these foci going to be? I mean, the spread of the virus is going to continue until it gets stopped. And so, something ultimately is going to block the spread of the virus. So the virus is really running a race with the immune system here. It's got to seed into the tumor and then amplify those infectious centers sufficiently to destroy the large part of the tumor before it gets stopped. So initial focus density, final focus size are the critical parameters. Next slide. So we, PhD student working in my lab actually did some really great mathematical modeling on this and came up with this approach, which may put people off who don't like math, but actually it's a really, really simple concept. So, the whole tumor has a volume that you can calculate, 4/3 pi r cubed. And the infectious center within the tumor has a volume you can calculate as 4/3 pi r cubed. And so if you then calculate the probability of tumor cells surviving in a tumor that has a single infectious center, it's going to be easy to calculate. Just take the volume of that infectious center, divide it by the volume of the tumor, and if it's a big infectious center, maybe it'll be covering 50% of the tumor, and so you got a 50% survival. If it's small, it's going to cover 0.001% of the tumor, and that's the probability of survival. And then what you do is you summate, can I have the next slide, please, Gail? You summate this. So you take the probability of survival for a single focus is 1 minus r cubed over r cubed for the small and the large foci. And then you raise it to the power k of however many infectious centers you've got, and you get the probability of the tumor cells surviving the infection. And if you use that very simple formula and you apply it using the table down below, you can see that if you have an ultimate focus radius of 12 cells, so that's a decent sized focus, and you only have 1 in 100,000 cells infected, you're not going to cure the tumor. Probability of tumor surviving is 0.99, 99%. If you increase the number of foci, we've gone here to 1 in 33,000 cells, 1 in 3,300 cells. You don't really get anything good until you get 1 in 3,300 cells infected, and then your survival probability is 0.05, so 5%. So you got a very high probability you're going to hurt that tumor. And then if you double the size of the focus, and that moves to the 25 cell radius, then you can see that 1 in 33,000 cells gives you a really good treatment outcome. And if you increase that number just threefold, then you're almost guaranteed that you're going to destroy the vast majority of the tumor cells in the tumor. So this sort of mathematical analysis, I think, really helps to illustrate what can happen if you elevate the size of each of these foci and if you elevate the number of foci in the tumor. So next slide. Yeah, that you just skipped it. Can you go back one? Yeah, this one. So this is just taking that model and plotting it. So the top of this waterfall is no response, and the bottom of the waterfall is a cure. And you can see that as you increase the number of infectious foci, and as you increase the number, increase the size of infectious foci, and you increase the number of infectious foci, you have the two interacting with each other to increase the probability of cure. And so that's essentially what we're after. Next slide. So what prevents us from getting a cure, assuming this is how it all works? Well, first, considering delivery, there are some key barriers to delivery that we know quite a lot about. The first is that if you put and I'm talking about systemic virotherapy here, as opposed to intratumoral, which is altogether a different game, and I'm not really addressing that in this lecture. But there's neutralization by antiviral antibodies in the circulation. If you send the virus into that kind of war zone, it is not going to reach the tumor. Also, if it's highly sensitive to complement proteins, it's not going to get to the tumor. You then have the problem of the virus being sequestered in the phagocytes, in the liver and spleen and bone marrow, in the sinusoids. And generally, if you put nanoparticles into the bloodstream, 99.9% of them are likely going to be sequestered in the liver and spleen. So if we could do anything to block sequestration, that would really help us with delivery to the tumor. And then comes the problem of extravasation, because the virus passing through the tumor blood vessel does not have any idea what's on the other side of that blood vessel. It just has to leak out of the blood vessel in order to find the tumor cells. So tumor neovessel permeability is a critical parameter driving the likelihood of success following systemic virotherapy. And you know, it may be an all or nothing thing, or it may be heterogeneous between different areas within the tumor. Next. So this is just to show the importance of antiviral antibodies, and we don't need to spend time on this, but if we passively immunize mice with anti-VSV antibody, then the therapy no longer works. Next. And if we then look at this heterogeneity question of viral extravasation in tumors, you can see here in the rotating image, in both rotating images actually, you can see the patecnate imaging studies for NIS expression done on an animal that's got a subcutaneous tumor and has received an intravenous dose of virus. And on days 2 and 3 post-virus administration, you can see these, this is the same animal, by the way. You can see the foci are becoming increasingly apparent, but you can see it's lumpy, and you can see there are parts of the tumor that are not well infected, as well as parts that are. So there's a heterogeneity problem here. Next slide. And that's illustrated here with the staining of tumor sections to show that there are fairly large areas with which we call voids, where the infection has not seeded and has not spread into. And so we've done quite a lot of modification of our mathematical approach to include voids. And we've been working on strategies that can address this problem of voids and increase the uniformity of delivery throughout the tumor. Next. So that's the delivery. Then we have to think about barriers to intratumoral spread. And because once the virus has arrived in the tumor, you know, the focus size is critical. And so what's going to constrain that? Well, the first thing is that the tumor may already be in an antiviral state when the virus gets in which case that virus infection is not going to spread even from the initially infected cell. If the tumor is not in an antiviral state, it can respond to the viral infection, so it can establish an antiviral state after it's been infected, and that will reduce the size of the foci. And then there are various cellular innate immune responses, such as the infiltration of NK cells, neutrophils, macrophages into the tumor, which can also impact the ability of the virus to spread. Then comes, after the innate response, then comes the adaptive immune response with cytotoxic antiviral T cells and antiviral antibodies. That's going to occur a little bit later, but if you've got a tumor that's allowed the virus to amplify, it's going to then be blocked and stopped by the adaptive immune response. And of course, there's always this question of hard tumors versus soft tumors, and the extracellular matrix being potentially capable of retarding intratumoral virus diffusion. So the next slide is just to illustrate this point that spread does not occur in the same way and in the same degree in every tumor. And these are some mouse tumor models where we gave the virus systemically. You can see there are plenty of little foci in the tumor, but they are not getting bigger. They've been shut down early on. And so the virus is, it's much harder to get an effective viral therapy in this kind of tumor. Next. And we needn't dwell on this. You know, everybody shows a similar slide when they're talking about the innate immune interferon responses. And this is just to illustrate that the viral infection itself turns on a whole bunch of antiviral genes, including the production of interferon, which further stimulates antiviral immunity. Next. And so, you know, if we look at this innate adaptive immune response, which is really the biggest constraint on virus amplification, you can see that early it's the innate response, and then late it's the adaptive response. So that's just to make that point. Next slide. Okay, so coming back to then the constraints on the boosting of the anti-tumor immune response, which occurs after this oncolytic stage of the infection. You know, I think that's going to be familiar to everybody listening to this lecture, that there is an immunosuppressive tumor microenvironment in most tumors, which is an important factor allowing tumors to survive in the face of potential anti-tumor immune response. So what can we do to address that barrier outside of just infecting tumor cells and driving the inflammatory killing? Next slide. And the obvious answer is we can put genes into the virus. We can modify the virus to make it more likely to provoke an immune response. Now, that's clearly a double-edged sword based on what I've just been telling you about the immune response constraining the spread of the virus. So now we kind of want to have our cake and eat it. We want to make the virus better at provoking the anti-tumor immune response. So which of those is the most important goal for us? We don't really know at this stage. We don't know what the balance is between the long-term immune control and the initial tumor destruction. You know, is it better to destroy a small number of tumor cells and then have a massive immune response? Or is it better to destroy a large number of tumor cells and maybe not have such a great immune tail on it? But there's many different genes that can be and are being inserted into all viral platforms to enhance the immune component of the oncolytic infection. Next slide. And that was the rationale for interferon beta. You know, we know that interferon beta in our virus can help the therapy. Interferon beta can be toxic to tumor cells. It's anti-angiogenic. It inflames the tumor and it activates immunity. It's the master link between the innate and the adaptive immune responses. But because all of the tumors that we treat are going to have variable susceptibility to the virus, the amount of interferon beta released following an oncolytic infection is going to depend on that susceptibility or resistance of the tumor to infection. And so hopefully interferon beta can be a leveler and can give us more responsive tumors than would be the case if we didn't have it in the virus. Next. So we did some studies in that 5T-GM1 model that I've been showing you, where we used VSV encoding an inactive interferon beta. So same virus, just the interferon beta doesn't work in mice. And you can see that although we get a nice oncolytic control of the virus in that right-hand panel here, the tumors come back. So we lose our long-term immune control when we take the interferon beta out of the virus. And we believe that the mouse interferon beta is actually enhancing the anti-tumor immunity. Next slide. Because if we deplete the animals of their CD4, CD8 lymphocytes, then we get a much worse tumor control rate. And if we take animals that have been cured following virotherapy and we re-challenge them with the same tumor cells, they are protected in contrast to naive animals. So we do think the interferon is really making a big difference to the immune response. Next. So the just briefly reviewing where we've got to here, we've reviewed the two-step action of oncolytics. We have looked at the anatomical, geometric, and mathematical aspects of oncolytic virotherapy using this example. We've identified the main barriers to systemic delivery and intratumoral spread. And we've considered the approaches that have been taken to virus engineering to impact boosting of anti-tumor immunity. And we've really skated over that. And, you know, there's clearly a huge amount of science in, you know, each one of these areas that you can dive into. So does it actually work in the clinic? And I'll just give you a brief snapshot of that. Here's a clinical trial that is ongoing at Mayo Clinic, in which we've seen durable responses at the top dose level. So if we go up through these dose levels from 5E9, 1.7E10, 5E10, 1.7E11, we eventually reach a dose where we're seeing really interesting responses. And if you just go to the next slide, here's a patient who had a durable complete response at the top dose level after one intravenous dose. She had quite treatment-resistant T-cell lymphoma. All these lesions on her legs are actually fat, juicy lesions that contain a lot of tumor cells. And you can see from the photo, 3 months after a single intravenous dose of the virus, there's a complete remission. And that remission remains durable at her 7-month return visit. And the next slide is another patient with extensive T-cell lymphoma, which you can see here on the PET scan before treatment. And note especially the spleen in this patient is lighting up quite strongly because it's infiltrated with tumor cells. And then a month after the virus infusion, that scan is looking a lot better. The response meets criteria for partial response because there still are some enlarged lymph nodes, and that response is durable at the 3-month return visit and is ongoing at the moment. So next slide. So basically the point I'm making there, just with a brief snapshot, is it does work given the right circumstances, given the right tumor, given the right dose of virus. It can work, but it doesn't always work. And, you know, addressing that heterogeneity remains a really important goal. So to conclude, oncolytic viruses impact tumor progression in 2 stages: oncolytic killing followed by immune tumor control. Mathematical modeling highlights the importance of maximizing both delivery and spread of an oncolytic virus. There are multiple barriers to systemic delivery and intratumoral spread, and the efficacy is going to be maximal when these barriers are addressed. So you cannot ignore them. You have to pay attention to them. And there are numerous approaches to virus engineering that can be taken to enhance the boosting of anti-tumor immunity after oncolytic virotherapy. And there are many that are aggressively being pursued. So oncolytics, in conclusion then, are rapidly gaining traction in the field of immuno-oncology. And the next slide is just my world's best Lego set slide. You know, I encourage anyone who's interested in oncolytics to dive deeper, because it's the most fun thing you can do. You know, the world is your oyster. There are so many different things you can dream up and try and test and explore. The next slide is just my acknowledgement slide. There are many, many people who've worked for many years on this at Mayo Clinic, and I have a wonderful collaborative environment at Mayo Clinic. But I should say that of the work that I talked about today, much of it was done by Shruti Naik in my lab, and the mathematical modeling side of it was done by Amber Miller, who's now working somewhere else. And with that, I'll just move to the last slide, which has to do with questions, and that you can see a much better speaker next week, because Carl June is going to be presenting on engineered T cells, and I can tell all of you he gives a fantastic presentation on that topic. So thanks very much.
Thank you very much, Stephen, for this excellent and very, very interesting lecture. So, and while we are waiting for the questions from the audience, and please, if there is any questions, comments, or so far, please just type it in. I can read it down to Stephen. So let me have the honor to ask the first one. So what about combination of oncolytic virotherapy, for example, with checkpoint inhibitors?
Yeah, that's huge. And you know, when I talked, and I didn't even mention it, but you know, when I was talking about engineering viruses to enhance that immune tail on the oncolytic virotherapy, the combination of oncolytics with checkpoint inhibitor antibodies against PD-1, PD-L1, and CTLA-4 is really moving very rapidly forward, because then you don't really have to engineer the virus. You can just add in that checkpoint inhibitor to prevent the T cells from becoming exhausted, and that gives you the same kind of thing that you might achieve through engineering the virus. The one approved oncolytic in the US is TVEC. It's a herpes virus developed by Rob Coffin, who was originally at University College in London. And that virus encodes GM-CSF, and it's been approved for the treatment of melanoma. But Amgen acquired the virus and then showed in a large randomized clinical study that if you treat melanoma patients with IPI, the anti-CTLA-4 antibody alone, versus with TVEC, the response rates are much better in that combination arm. So TVEC and IPI are definitely complementing each other pretty well. And there's a big trial going on, which we're all anxiously awaiting the readout from, of TVEC plus Pembro, because the data from that combination in the phase 1 study were considerably better than TVEC plus IPI. So yeah, that's coming. And we're testing VSV in combination with cemiplimab, which is Regeneron's anti-PD-1 antibody at the moment as well.
Thanks. We have received 2 questions from the Lund University, and these are from Christina Pires. Thanks, Stephen, for the nice talk. Perhaps I lost it, but what makes the VSV virus target specifically the tumor site after IV delivery? This is the first one.
Yeah, it's the interferon responses of tumors are in general somewhat defective. And the VSV is quite highly responsive to the antiviral defenses that cells mount when they've been treated with interferon. So our virus is therefore able to spread in tumors where the interferon response is impaired. and where the microenvironment is immunosuppressive. And they pump interferon into that environment, which allows the immune system to come in. But in a normal tissue infected with this virus, there's a very strong interferon response. That means that the initially infected cell may release progeny viruses, but they're not going to infect the surrounding cells that have already been exposed to interferon.
Thanks. The second question from her is another question. What is the limit in terms of cargo for introducing additional genes?
Well, in VSV, the limit is probably reached in the virus that I showed you with VSV, interferon beta and NIS. And in that virus, we've added about 4 or 5 kilobases of additional sequence. I think going beyond that is possible, but the viruses get longer and longer and less, they grow less and less efficiently. So in general, with oncolytic viruses, you do lose something in terms of efficiency of propagation when you modify the genome. The capacity for foreign genes is larger in larger viruses than it is in smaller viruses. And one of the advantages of the herpes simplex virus system, which is about 200 kilobases before you start playing with it, is that it can take quite a lot of additional genetic material, as can vaccinia and pox viruses. But these are much more complex viral genomes, and I think for the purposes of the lecture today, it was I think more helpful to go through the simple genome of the VSV.
Thanks. So we have received 3 questions from Margarita Romanenko from the University of Minnesota. Thank you for the great lecture. I have several questions. First, what is the limit of the VSV genome capacity for the transgene? So maybe you have already, you have basically answered this, right? Yeah. And is the IT delivery better option in comparison to the systemic if the virus is highly immunogenic and evokes anti-tumor immunity?
So the important data that persuades me that systemic will be the game changer in oncolytic virotherapy comes from the registration study that was done with TVEC, which is, because it's a herpes virus, it can't be administered systemically. And TVEC is given by intratumoral injection. And in the phase 3 trial that led to the approval of that agent for treatment of melanoma, injected tumors responded very frequently. Distant cutaneous tumors that were not injected responded less frequently. About 22% of those disappeared in treated patients. And then deep visceral lesions were even less responsive. And about 9% of deep visceral lesions did show a complete response. So if you get the virus into the tumor, you have a much higher probability of that tumor responding. So while it's clear that you can boost anti-tumor immunity by intratumoral injection and you can have a systemic effect, it's going to be way superior if you can efficiently deliver via the bloodstream. In the world of immuno-oncology, we know that the checkpoint inhibitors work, and we know that cell therapy can work. We however, also know that hot tumors respond much better to checkpoint inhibitor therapy. And so the goal of immuno-oncology broadly is to generate some kind of agent that can be given systemically that can turn cold tumors hot wherever they are. And I think that's where we're headed with oncolytic virotherapy. There may be other approaches to this that are going to be successful, but I think oncolytic viruses are probably leading the charge at the moment in that regard.
The third question of Margarita is, how did you overcome virus clearance in your patients?
Simply by increasing the dose and accelerating the infusion. So we administer our virus over a 30-minute infusion. which is quite rapid given the number of virus particles that we're administering. And the dose, just so that you understand, is very high. We're up above now 10 to the 11 as the virus dose. The dose of virus administered when TVEC is given by intratumoral injection is 1 million infectious units for the first dose, and then 100 million for subsequent doses. And the titer does not, the dose does not go above 1E8. So we're 1,000-fold higher than that going intravenously, because our virus is immediately diluted into a large volume. And we need to get a high enough concentration, at least transiently, to drive that virus across the tumor neovessels into the tumor. So that, I think, is the key. And by using VSV, we know that none of our patients have got antibody titers against the virus at baseline. Similar work that we did with measles virus ran into a brick wall, because most patients do have anti-measles antibody. And that really does preclude the effective use of intravenous measles virus as a therapy.
I was amazed how fast you have seen the virus spread in your pictures. And when you now, you also explained the barriers with regard to innate and adaptive immunity. So can you give us a little bit a timeline which we have? So of course, innate is quite fast, but how much time do you have, or the therapy has, before the adaptive immune system kicks in?
Well, we can detect neutralizing antibodies against the virus by day 7. And so when you administer a virus intravenously, I think that adaptive immune response is starting to develop straight away. I don't know at what time point it really becomes important, but repeat dosing of the virus, even on day 4, does not further increase the efficacy of the therapy. So we think that it's fairly rapid. Now, it probably varies greatly from virus to virus. VSV, I like to call a hit and run virus. That virus comes in, it does its work super fast, and it goes, and you're not going to come back and use it again. So it's a single shot therapy, and it needs to achieve what it's going to achieve with that initial dose. And that can improve the response rates to checkpoint inhibitor antibody as well. So, you know, that's a real nice combo for the virus. Viruses like herpes virus, the TVEC, they hang around. And if you just think about herpes infections that naturally occur, cold sores keep coming back in people who have extremely high anti-herpes antibody titers and extremely powerful anti-herpes T-cell responses. And many of these patients in the TVEC trials do have, ahead of therapy, powerful antiviral immune responses. You know, that's one of the reasons it can't be used systemically. But the virus nevertheless hangs around in the tumor and does something, because it really does effectively combat that innate immune response. So those are two ends of a spectrum, and there's a lot in between of viruses that have different rates of propagation and different susceptibilities to the innate immune and adaptive immune defenses.
We have received a question from somebody from the Technical University in Munich, and he asked, or she asked, did you ever see neurotoxic side effects by the virus in humans? And it also said, thank you for the amazing talk.
Okay, so neurotoxicity was a major concern at the outset. VSV has been studied extensively over many, many, many years, and it's well known that if you give it to immune-compromised mice or rats, it will be fatal, and that will be a consequence of neurotoxicity. If you inject it into their brains, it'll be fatal due to neurotoxicity. However, it doesn't cause neurotoxicity in cattle, where it naturally infects. And we've seen no evidence in our human studies of any neurotoxicity. One of the modifications we made to the virus, which reduced its neurotoxic potential in mice, was the insertion of the interferon beta gene. But that didn't completely abrogate its neurotoxic potential in mice. And if we put this virus into the brain of a mouse, it will cause neurotoxicity. So there's a big kind of species difference there, which was a barrier to the initial clinical translation, and which, you know, raised a lot of questions from FDA, meant we had to do non-human primate studies in order to justify moving into human. So it's an important question, but no neurotoxicity to date.
Thanks. I don't see any further questions, so I really would like to thank you again for this fantastic talk. And as you said, I would like to invite our audience to see or visit us next week when we have a talk of Carl June, which is entitled Updates with Engineered T Cells. So thanks again, Stephen. Thank the audience for being with us, and have a nice day. Bye-bye.
Thanks very much, Hildegard. Bye.